I'm David. I'm a prebiotic chemist, really more of an organic chemist. And in Becky's introduction talk, she reviewed chemical structures for you. So I hope you got that, because otherwise it's going to be a little rough ride. But here we go. All right. So fundamentally, life is a chemical phenomenon. I don't mean to be patronizing with this. I just want to highlight that, you can see a schematic of DNA, but it really does have fine chemical structure, and that fine chemical structure is important for elucidating the origin of life. So, in experiments like the Miller-Urey experiment, which we're all familiar with, typically the thing that you find are amino acids. So, for example, the constituents of a protein. But if what we're trying to figure out is how another class of biomolecules, the nucleic acids, came into existence, the monomers of nucleic acids are comparatively more complex. So compare an isolated amino acid to an isolated nucleotide, and clearly the nucleotide is larger and more difficult to assemble in a prebiotically plausible manner. So before I jump right into my chemistry, I wanted to give you a little bit of an overview of how biology works today, because that's how we are informed of what molecules to target in our prebiotic syntheses. So DNA, another schematic. DNA is the hereditary molecule of all life, with the exception of some viruses. DNA is transcribed by a protein into an RNA message, and then that RNA is translated by an enzyme called the ribosome, which emphatically is not protein-based, it's RNA-based, and that gives you proteins. But you require proteins in order for DNA to replicate itself. So because of this, we have a chicken and egg problem, right? We need proteins for DNA, and we need DNA to code for proteins. So how do we get out of that cycle? Well, there's a clue here in the central dogma of molecular biology, which is RNA. RNA in the central dogma fills two different roles. One of them is informational in the form of messenger RNA, and the other one is catalytic in the form of the ribosome, which itself is a ribozyme, an RNA enzyme. So since RNA has the capability to perform both these functions, which today are mostly partitioned between DNA and protein, it's thought that RNA played a really critical role at or close to the origin of life. All right, so that leads to this popular idea called the RNA world hypothesis, which is, but before I get into that, I want to explain to you exactly what RNA is and how it's distinct from DNA. So here's that schematic again, and here's the fine structure again. RNA is very similar. So if you look at DNA, you have this phosphate deoxyribose backbone with the nucleobases here. RNA is similar, but there are just a few modifications. So in DNA, we have the nucleobase thymine, and in RNA, we have the perhaps simpler nucleobase uracil, which pairs with adenine. DNA has a hydrogen atom here, where RNA has a hydroxyl group. Now, superficially, that might seem to make RNA more complicated, but actually, that hydroxyl group is easier to produce in a prebiotically plausible manner than the deoxyribose of DNA. And those small modifications have profound consequences in what RNA and DNA are able to do. So DNA is optimized for information storage and transfer. RNA can perform that function too, but because of this hydroxyl group, there is a change in the bulk structure of RNA, which allows it to more readily access catalytically active structures. All right, so now we're at the RNA world hypothesis, which is the idea that before the DNA protein-based biochemistry of today, we had a biochemistry that was predominantly based on RNA. I just want to really quickly note that that idea, as I've just stated it, has been contested a bit lately, but let's just stay within that paradigm for now. And I'm going to give you a historical perspective on how it's been investigated. So what we want to get to is this polymer, RNA. The traditional approach has been to start from simple prebiotic precursors. Sorry, there's a little bit of feedback. The historical approach has been to start from simple prebiotic precursors and build your way up to the polymer. Now, if you draw it like this, it looks pretty reasonable, right? And this is, maybe you've heard of the term paper chemistry. You can draw it on paper and it looks really pretty, but it's completely wrought with issues. So for example, if we want to get the sugar ribose exclusively, that's a big problem because the chemical reaction that's typically invoked in order to produce sugars in a prebiotically plausible manner is totally non-selective and produces a variety of sugars. But even if we could produce ribose exclusively, we have a further problem in that the canonical nucleobases, A, U, G, and C, do not react with ribose in water in the absence of enzymes to form nucleosides. Even if we could form nucleosides, though, there are difficulties in phosphorylation, and I'm sure some of you are aware of attempts to overcome these difficulties, which indeed do phosphorylate organic compounds, but there remains the problem of polymerizing those organophosphates to get this phosphodiester polymer. So, we in the HUD lab and in the Center for Chemical Evolution in general have this new approach, which is basically the structures that are present in life today are not necessarily those that were present at the very origin of life, and that applies to RNA as well. So if you're willing to accept the idea that DNA is the descendant of RNA, perhaps RNA is the evolutionary descendant of a series of pre-RNAs going all the way back to this Proto-RNA. It's an older nucleic acid, but it checks out. And so what we do is we take the nucleic acids of today and we partition them into 3 structural components. So one of them is the ionized linker, which today is phosphate. One of them is the trifunctional connector, which today is either ribose in RNA or deoxyribose in DNA. And then we have the recognition units, which are the canonical nucleobases, A, U, G, and C. And these structural components, we think, would still be present in the most ancient nucleic acids, but they might have been replaced with different chemical moieties which had chemical properties more amenable to spontaneous formation than this phosphodiester polymer. Okay, so first I'm going to focus on the recognition units of RNA and how we can start to speculate on what may have preceded the extant nucleobases. So if you have a prebiotically reasonable milieu of nucleobases, some of them will be the ones that are present in life today, but surely just out of the natural trajectory of prebiotic chemistry, you're going to have a bunch of other heterocycles that are not featured in life today. So how do we sort out from that complex mixture a set of nucleobases that can form an informational system? Well, let's use the concept of supramolecular assembly to our advantage. Some of these heterocycles will have the ability to spontaneously self-sort into a supramolecular aggregate, and they'll exclude all the other nucleobases. Once we have that non-covalent supramolecular assembly, perhaps we can use that as a scaffold to create a covalent polymer. So these are the heterocycles that we're most concerned with. They have the ability to form these supramolecular structures by having 2 hydrogen bonding faces. So what that means is that they have these edges that have very polar sides, and they snap together with their pairing partners to form these hexad structures. So the bases that we're concerned with are barbituric acid, triaminopyrimidine, or TAP, as I'm going to refer to it in the rest of the talk, cyanuric acid, and melamine. They all have the ability to form this hexad, which forms spontaneously in water. And the really cool thing about this hexad is that once it's formed, it presents a very large hydrophobic surface in water. And that is very unfavorable to present to bulk water. So what it does is stack one on top of the other in order to hide those hydrophobic faces, and you have this supramolecular assembly as a result. All right, so we have these non-canonical nucleobases that we want to form polymers out of. Do they actually react with ribose in order to form nucleosides? Turns out that they do. This is work from 2013 from Michael Chen and Brian Cafferty in the HUD lab. So if you take this heterocycle, TAP, and react it with ribose, there are a variety of products that you can form. And the details of this are not important, besides that the one you form in the greatest yield is the beta-ribofuranoside, which what that really means is it has the same conformation that nucleosides in life today have. So the problem is that if you had ribose, there were all these possibilities, and it's not immediately clear why we have this one in life today. But this suggests that it's just a natural outcome of the chemical reaction. And if you take this nucleoside, which we call TARC, and you incubate it with cyanuric acid in the appropriate buffer, you get these supramolecular assemblies, and you can even visualize them by atomic force microscopy. You can see here that they have the appropriate diameter for what we predict this supramolecular assembly to be. All right, so that's cool, but if you want to have an informational system, you need to have at least 2 units. So just having TAP is not enough. So what we're going to do is take inspiration again from extant biology and make inferences about what extinct biology could have been like. So we have the AU base pair, which cannot form spontaneously in a prebiotically plausible manner, remember that. But perhaps we can have a similar base pair, which is structurally analogous, but that can form spontaneously in a prebiotically plausible manner, just like TARC. And it turns out that that is indeed the case. So barbituric acid and melamine both have the ability to react with ribose in water, ribose 5-phosphate in this case, to form nucleotides. And they have the ability to assemble, just like before, at the appropriate pH, to form the supramolecular assembly. And again, you can see this by atomic force microscopy. And another really cool feature of the system is that these fibers are so long and so interlinked that they prevent the bulk flow of water, so you get this hydrogel. So that's a cool material property, but what's really important here that I want you to take away is that we have an information system now, as long as we can figure out a way to polymerize them, because we have 2 units. So really quickly, I want to tell you about another component of RNA, which is the trifunctional connector. So just now I was telling you about how we're not going to deal with the canonical nucleobases because they don't react with ribose in water. But if we're willing to accept that the nucleobases may have been different, we should have that exact same attitude with all the other components of RNA. So for example, we're going to take the trifunctional connector, which today is ribose, and see if it could have been something different. Now, I mentioned before that it's really difficult to get ribose in a selective manner because the reaction that typically is invoked to produce sugars, the foremost reaction, is inherently non-selective. It produces a ton of sugars. Like, there's a chromatogram that was shown in Becky's slides, I think, that shows the products of the foremost reaction, and there's like 100 peaks. There's even more than that. So we had this question. If you do naturally produce a ton of sugars in these prebiotic reactions, can a non-canonical nucleobase react not just with ribose, but perhaps a suite of them to form non-canonical nucleosides, or glycosides, really? Now, again, chemical structure is terrifying, but all I want you to take away from this is that there's a variety of sugars which ostensibly have different chemical properties and may or may not react with TAP. So what I'm going to do is I'm going to take a general sugar, just read this word, and react it with TAP, and you can get a bunch of different products. And it turns out that it works. So in this chart, I have the yields here. So we did this either at pH 1 or pH 7 because it turns out that this reaction happens to be acid catalyzed. But I want you to note that on every single structure here, the reaction worked in at least one of those conditions. Every single one. And that's important because it's often just assumed without any kind of, it's assumed a priori that ribose was first because that's what's in biology today. But that's not necessarily a good assumption because, at least from a reactivity standpoint, there's nothing special about ribose. All of these sugars, all of them, have the ability to react with a non-canonical nucleobase. Okay, great. So now this is going to really be for the chemists in the room. We wanted to take a closer look at the structural outcome of some of these reactions. Now, it turns out that glucose is one of the easiest sugars to study because it's one of the most well-behaved and structurally congruent. So if we take a glucose derivative and react it with TAP, there's basically 6 possibilities. Up here, these all have the TAP nucleobase pointing up. That's called beta. These all have the TAP nucleobase pointing down. That's called alpha. The reason that's important is because in life today, almost all glycosides of nucleobases are beta. And it turns out that when we react TAP with glucose or a glucose derivative, all of the products are beta. So that's very interesting. But that might be intrinsic to glucose derivatives. Nevertheless, when you get the reaction of TAP with glucose, you get every possible substitution on the TAP molecule because TAP turns out to be a multidentate nucleophile, so you can have different positions of substitution. Glucose 6-phosphate behaves the exact same way, and strangely, N-acetylglucosamine does not tolerate the C substitution. This is a really esoteric point, but that might have to do with diminished electrophilicity of the relevant intermediate in the reaction. Okay, so supramolecular assembly again. Is it possible that these non-canonical nucleosides, which do not feature ribose, have the ability to assemble in water? Well, I took one of them and I purified it, just because it's easier to study that way. When you incubate it with cyanuric acid at the appropriate pH, it forms assemblies, but they're really small. This is, again, an atomic force microscopy image. That's weird. But what if I take the crude reaction mixture? So what that means is I perform the reaction and I took the reaction without any purification and mixed it with cyanuric acid. Those form very long fibers, which is interesting because this is the more prebiotically reminiscent scenario. So maybe that's telling us something about how these polymers got going. Okay, really quickly, I just want to point out that in this study, we had an interesting result, which is that a sugar called ribulose, we anticipated to react with TAP in this manner to form these strange nucleosides, but it turns out that there's no evidence for their formation. Rather, it forms these arabinosides and ribosides. That's cool, because ribosides are nucleosides. Those are the things that are present in life today. Now, why are we excited about this? I told you earlier that it's really difficult to get to ribose in a prebiotically plausible manner, but it turns out that ribulose is a little bit easier. There's a synthesis by Ram Krishan Murthy where you can get to ribulose, and in our system, we think what's going on is that it's isomerizing in situ to ribose and arabinose, and then reacting with TAP to form these nucleosides. So that's pretty cool. This is the proposed prebiotic synthesis of ribulose. And if you want to learn more, please see Tyler's poster, which is all about this. Okay, so just to recap, we are taking the stance that the structures that are present in life today are not necessarily those that were present at the origin of life, and most importantly, RNA was not emphatically first. So if we take this approach, we get some really interesting results. The nucleobases could have been different. We go to ones that are more reactive and have a greater propensity for self-assembly. The sugars could have been different, and that sorting that out that's going to be fun. I didn't talk about the ionized linker, but we are investigating that in our lab, and if you want to learn more, just ask me later. Okay, so I just want to thank everyone who made this possible. Brian Cafferty was my mentor when I first joined the lab. Kim Clark did all the atomic force microscopy that you just saw. Tyler's our new graduate student who's taking on all this ribulose stuff. I was really lucky to have really talented undergraduates, Catherine and Megan, and really great mentors, especially my PI, Nicholas Hutt. So thank you all.
All right, we have time for a couple quick questions.
Hi, great talk.
Thanks.
In your part with interaction with TAP and barbituric acid, do you think that the interaction between TAP and barbituric acid and those ribose phosphate derivatives, is that dependent on their inherent chirality as well?
Could be. So you're talking about the chirality of those nucleosides as endowed by the sugar.
Yeah.
Okay, it could be. But we performed this study with commercially supplied sugars, so they are emphatically not racemic. They're all enantiopure. So we have not investigated that yet. But there is research in our lab, which, find me later, I'll tell you about it.
Yeah, that's the second part of that question, if it's fine, is that would it be possible then, or have you tried something like that, in which a chiral or one part of it resolves another which is racemic, or is that something?
That's someone else's work in my lab, and I can totally tell you all about it, but it's long.
Is that a question?
Hi, great talk. In general, glucosides, also something aromatic linked to a sugar, are very, very common in plants, which are used for all type of signaling and regulation. Do you think that if we focus just on RNA and DNA, we limit us in understanding, for example, ancient signaling pathways or reaction networks? Because these things, they might look more similar to what like plants and bacteria use nowadays.
Yeah.
Rather than the information system.
Yeah,
Or
I
just
think
just complementary information system.
Yeah, okay, so related to what you're saying, it's interesting to think about how so many cofactors in life are basically nucleotides, like NADH and stuff like that. I think that might bear a clue as to the origin of nucleic acids, but it, also possibly the origin of a more rudimentary biological system in general. But we can talk, that can go into many avenues, so I'll just leave it at that.
All right, so in the interest of time, we're going to move on, and let's thank David again.
Thank you.